Hello, everyone, and thank you for joining today's webinar on dual organ-on-chip model, featuring the case study on liver kidney organ-on-chip system to investigate tacrolimus disposition. My name is Anel, and I'm a life science application engineer from Fluigent. And we are pleased here to be joined by Dr. Nicolas Vedren and Izzy Petit, so associate professor and the PhD candidate from University of Limoges, the INSERM Unit 1248, Pharmacology and Transplantation. In today's session, we will introduce the key concepts of dual organ-on-chip models, the concepts and applications. I will also have a chance to give you a closer overview of a Fluigent Omi organ-on-chip platform designed to support long-term cell culture and control the microphysiological conditions. Then Izzy Petit will introduce a case study on tacrolimus disposition in liver kidney model, where the Omi system was used for dynamic perfusion. Finally, we'll have the perspective, the future outlook in pharmacology and transplantation research overview by Dr. Nicolas Vedren. And finally, I'll give you a technical overview, practical considerations for Omi use, and we'll open the floor for Q&A. Let me begin by introducing the different in vitro platforms that allows us to study different cellular and tissue interactions, and the difference in between different type of in vitro models. So in vitro models, they increase by complexity to have a closer physiological relevance here. So for instance, first of all, we have the 2D monolayer soil culture in Petri dishes. The classical models that were developed in the early 20th century, they offer simple but sometimes limited physiological relevance. It's also suitable for dual co-culture of different type of cell lines. Then we increase the complexity by having the transfer or permeable membrane systems that allow 2 layers of cells to be cultured separately and enabling diffusion of soluble factors, widely used for barrier models. Then with one step further in increasing the complexity, there comes a 3D structure embedded in hydrogels that could be tumoroid, spheroid, or organoids. And here, the extracellular matrix composition, the stiffness, permeability of the ECMs significantly influence the tissue function or tissue representation. And then we have a microfluidic 3D dynamic conditions, or culturing on a chip, where the systems allow to incorporate the flow, the shear stress, the compartmentalization of different tissue types or cell culture types via membrane separator or hydrogel separation to produce tissue microenvironment with a higher fidelity. So what you can see here, we increase the cell culture complexity to increase the relevance to the biological tissue as well. As we move from 2D to 3D to microfluidic systems, we increase dimensionality and the physiological relevance that allows us to have more predictive responses. Physiological relevance is not the only main reason to study in organ-on-a-chip systems and to increase the complexity by moving to multi-organ-on-a-chip systems. One of the main reasons are studying the drug dynamics, how they're processed and exchanged in human organs in a dynamic way. Currently, in drug development, animal models are mostly used for pharmacokinetics, for ADME assays, for metabolite-mediated toxicities. And in the context of global initiatives, like we hear a lot with FDA Modernization Act and the recent EU directives, there is a higher there is a higher interest in replacing the animal testings. And how it can be done? It can be done with organ-on-chip systems as well as its organoids to model inter-organ crosstalk, to have the isolated studies on pharmacokinetics and ADME modeling, and also to study the toxicity and metabolites. The dual chip systems or dual organ systems help isolate the specific contribution of each organ while capturing a crosstalk at the same time, making it a very powerful tool for mechanistic studies. And while we're talking on about the drug metabolism, we also always consider the important and central role that liver plays in this mechanics, in enzymatic clearance, in biotransformation, biliary excretion, and first pass metabolism. And this type of interorgan crosstalk can be designed on the microfluidics level to have the ordering between different type of organs such as liver gut, liver heart, and liver kidney to study respectively the first pass metabolism, but also the cardiotoxicity and the renal clearance in different models. So dual and multi-organ models allow this axis to be examined under controlled, isolated controlled models, providing the insights that static models cannot provide, especially when it involves a lot of communication with various tissues. When we talk about the organ-on-chip models, we also have to consider the perfusion, the role of perfusion plays in this inter-organ crosstalk. And perfusion and recirculation are important because they mimic the movement of these biomolecules between the organs, and it allows metabolites and drug byproducts produced in one organ to reach another by creating a more accurate interaction that biologists would like to study. And we have different approaches, different configurations that this can be done. So you can see here the conditioned media transfer, the dual compartment chips. Here you can see the image of a gut-on-chip model designed in Institut Pasteur Lille by the group of Alexandre Grassard. So there is two different compartments can be perfused at the same time or one at a time. There is also a manual conditioned media transfer. So for example, if you culture one type of organ on one chip, and then you can manually transfer the media to the second organ and culture long-term in the conditioned media. And there is also a way to interconnect chips and study the several chips in a sequential manner that transfer the medium. So how it would have been done on the human body. And all of this can be done with Omi. So you can either transfer the conditioned medium from one Omi to another, so one device to another, as illustrated here. There is also a way to have dual compartment in a chip perfused in a synchronized manner with two Omis. And as Izzy will later illustrate, there is a way to have a sequential connection of the chips and the Omi devices. So maybe I moved a little bit too fast. For those who are not aware of the Omi system or who are new to Omi system, let me introduce it to you. So the short overview of Omi, it allows the microenvironment control during the cell culture inside the microfluidic chip. It's a small device that primarily can be used in a single chip configuration, as you can see here. It's transportable and can be controlled via tablet to perform different type of fluidic functions. So for instance, we will focus here on recirculation. We'll place a medium inside the device and we'll recirculate the medium inside the chip. So first configuration is a single Omi. You can have one Omi device connected to one chip and recirculate it in a controlled way through a Fluigent technology based on the pressure. Then we have the dual Omi configuration where you can control two Omi in a synchronized way to have a liquid co-culture interface that is illustrated here. And we also can have the experiments parallelization that allows higher reproducibility. So for instance, up to 12 independent Omi devices can be controlled at the same time with one tablet, and it can be easily mounted on two incubator trays. And the great thing is that this modular designs is convenient for assessing each chip or each model independently for observation, sampling, and maintenance. So if it wasn't clear, we will return back to the how does Omi work and its functions. Please do not hesitate to write all of your questions in the Q&A panel. And now I would like to pass the floor to Easy Petit, who will guide us through the liver-kidney organ-on-chip setup established in their laboratory in Limoges and presents his finding from the tacrolimus disposition study.
Okay, so I will take the floor. So
go ahead.
I'm correct. This will work. So good afternoon, ladies and gentlemen. So I'm Easy Petit, a PhD student. And today I'm going to present to you a case study around tacrolimus disposition in a liver kidney organ-on-chip model. So first, a little bit of context. We are a pharmacological team, and we are particularly in our group interested in pharmacokinetics. So it's how a chemical compound or a drug is processed by a living organism. It goes through four main phases: the absorption, distribution, metabolism, and the excretion. And there is two organs that are particularly interesting. It's the kidney and the liver. The liver for the metabolism aspect, but also for the biliary excretion, and also the kidney for the urinary excretion aspect. And these two functions are almost mediated by xenobiotic transporters. And those are transmembrane protein that mediate the translocation of xenobiotic across cell membrane. There exist two main families, the ATP binding cassette and the solid carrier, and they exhibit some interesting feature. First, they are present in almost every living organism, but also almost all human organs, and they can interact with a broad range of xenobiotics, but also endogenous metabolites. And this feature places the drug transporter in a central place in inter-organ communication, in homeostasis regulation, and this is the kind of interaction between xenobiotics and endogenous metabolites that are particularly interested in. We choose the organ-on-chip technology to explore that. First, because it's well known that it improves cell function, it increases predictivity, the physiological relevance. It's particularly true in case of drug transporter, where the dynamic condition allowing by organ-on-chip enhance transporter expression. There is also the multi-compartment aspect. We speak about inter-organ communication, so that's really important. And one other aspect is the human relevant data that are particularly important in case of drug transporter because species specificity are really important. So first, we start by developing our kidney model. And to do so, we look at the nephron physiology and all the function that the nephrons do, so the filtration, the reabsorption, and the most important aspect that we would like to replicate is the secretion aspect, so the transporter-mediated drug elimination. And this is mainly due mainly due in the proximal tubule. And this place is really important because it governs renal clearance, drug interaction, but also drug-induced nephrotoxicity. So it plays a really central role in urinary excretion. And to replicate this place, we have chosen a commercial culture support, which consists of two compartments, basal one and an apical one, separated by a semi-permeable membrane. And we use this setup to model the blood flow and the urine flow, and to replicate the proximal tubular epithelial barrier, which is the hepatic TERT1 cell line. So we would like to replicate this place and to establish a proximal tubular organ-on-chip model that mimics drug transporter excretory function. To do so, we first need transporter, and yes, in our model, we express transporter, and more importantly, under dynamic condition, we saw increased expression, so our model better express transporter than under static condition. We have the transcriptomic aspect, we have the expression, what about the protein? And yes, we have the transporter protein. And more importantly, they are correctly localized in under dynamic conditions. So we have a robust cell polarization. So expression, protein, what about the functionality? And yes, in our setup, we exhibit a polarized transport, which means that our model recapitulates the transporter expression. This work has been published in early 2025. So, okay, we have a PT-on-chip model, quite good. Now, go to the liver. So why the liver? Metabolism, excretion, really important aspect, notably that the excretion into bile, but also back into recirculation. And it's really important because it plays a central role in detoxification, metabolism, biliary elimination, etc. So it's a really important organ. And to replicate it, we chose another culture support, commercial culture support, that allow us to culture spheroids under blood flow or under dynamic condition. And we choose the HepaRG cell line to generate those spheroids. One key aspect of the HepaRG is the dynamic expression of cytochrome, so metabolizing enzyme. And this dynamic expression is well known in 2D, but in 3D, it's not so well known. To look for that, we make a time course evolution of CYP3A4 and CYP3A5 mRNA across HepaRG differentiation in spheroids. And we saw two time points where the expression was higher, and we choose to develop from that this cell culture protocol. So at day 0, we start differentiation in spheroid model, and we can put them under flow shear stress. So under dynamic condition, start from day 7 and treat them at day 8. We can maintain those spheroids under a dynamic condition for more than 5 days. If there is no bubble, there is no problem. And we also look, so we have the mRNA. What about the protein? Yes, we have metabolizing enzyme, but also drug transporter. And also, what about the functionality? And yes, we have metabolism. We choose a classical probe, which is midazolam, and we treat our model with that and see the apparition of metabolite, which means that our model can metabolize midazolam. So, okay, we have a proximal tubular on-chip model, a liver on-chip. Now it's time to combine them into an integrated dual organ-on-chip platform. And with this dual platform, like you can see, we have an hepatic compartment that goes to the kidney, a urinary compartment that goes also to the kidney, but at the apical part. And can we track drug metabolism and clearance? For the metabolism, okay, no problem. We have the apparition of the metabolite, and for the clearance, no problem also. We have the apparition of midazolam and the apparition of the metabolite. So that's a proof of concept, works quite well. But here comes the problem, the tacrolimus. So tacrolimus, briefly, it's an immunosuppressant that is widely used in organ transplantation. It undergoes hepatic metabolism by CYP3A4/5. It's mainly eliminated in the bile, but it can go at the kidney and generate nephrotoxicity, and it's also a little bit goes through urinary elimination. But the real problem is the highly lipophilic. It's an highly lipophilic compound that gets adsorbed everywhere to every surface. With our previous setup, we have more than 70 to 80% of the doses that just vanish. But if we have a problem, we have a solution. And our solution goes with the Omi setup. So Omi, it's an automated flow. It has an automated flow control. It miniaturized. It's compact. We can take it under the biosafety cabinet. We can take it under the microscope. So okay, that's great for us. But the real important aspect was the reduced working volume that enable the metabolic accumulation. And there is also this low surface to volume ratio that's lead to tacrolimus absorption much easier to manage. So we have this dual organ-on-chip setup with the liver on chip, the PT on chip, and two Omis to drive them. And we treat with tacrolimus, but also we are interested in tacrolimus plus metformin, this DDI, and we treat our hepatic compartment with these two cocktails. So can we follow drug metabolism and drug excretion, particularly tacrolimus? And yes, at the hepatic compartment, we saw if we look for tacrolimus, we saw rapid decrease due to liver uptake, but also due to adsorption. And at the urinary compartment, we saw the apparition of tacrolimus, which means that we have a clearance, the excretion of the tacrolimus compound. And it's at quite low doses, which could replicate the physiological aspect with this low urinary elimination. So the clearance, that's okay. And what about for the metabolism? And yes, we have the apparition of the main metabolite, so the desmethyltacrolimus in the hepatic compartment. And we only saw traces in the renal compartment, in the urinary compartment. But okay, our model can reproduce metabolism and renal clearance of the tacrolimus. But we would like to push further. To do so, we have used targeted metabolomic analysis to look for 103 metabolites. And by PCA, we see that there is separated group depending on the treatment, notably at the urinary compartment. And that's almost the case for the hepatic compartment, especially true for the tacrolimus and metformin. And there is also a compartment-specific response. If I take the example of creatinine, we saw that at the hepatic compartment under tacrolimus treatment, we have an accumulation of creatinine in this compartment, in our blood hepatic compartment. And at the urinary compartment, it's the inverse. So in the control, we have higher dose of creatinine and lower under tacrolimus treatment. And creatinine in the blood compartment suggests an initial loss of the renal function. And by adding metformin at the hepatic compartment, we have a decrease in creatinine concentration. That's for the hepatic compartment. And for the urinary compartment, we have only a slight increase. And this slight increase could be due to the competition between metformin and creatinine for SLC47A1 and 2, which are drug transporters. And this is the kind of interaction that we are really interested in. This is the kind of discussion that we would like to really push further. And with those that's only preliminary result, but that's what we would like to push further. And this is done thanks to the Omi setup that help us to track drug metabolism and excretion in a separated compartment. We can also perform metabolomic analysis. And well, I didn't really show that, but we can perform classical assay, mRNA, immunolabeling, and we have shown that in the application note. But this also came with limitation, so it's longer to start up and shut down experiment. It's technical time, it's a little bit longer. Everything is automated, so there is reduced operational flexibility. We also have occasional technical issues like with every microfluidic devices. And if I'm correct, we were the first to really play with the dual Omi specification like that. So the software might need a little improvement to really meet the dual Omi specification. But it also came with much higher advantages. The first one is the miniaturization. So it's portable, practical. We can put that under the safety cabinet, microscope. So that's really good, really great. There is also the reduced media volume. That's really important for us. The automated single sampling. So we can program automated sampling, which means that we can access to data point during night. There is also this continuous recirculation. So globally, if the launch, if everything goes well, you just launch your experiment and it works perfectly. So that was really great. So that will be quite all for me. So I would like to thanks So I would like to thanks all of my coworkers, but also a special thanks to Annelle and Antoine from Fluigent and every one of you for your attention. So thank you, and I will be pleased to answer to every of your questions. So I think that's okay for me. So I don't know which, I think it's Nicola that will... Annelle, I don't...
Hello.
Oh, that's okay. I hear you now.
Okay, super. I was saying thank you, Izzy, for a detailed overview of your experiments and to share how you establish different compartments in organ-on-chip model and your results. Thank you for your time as well for passing on the dual organ-on-chip model and to help us also test the Omi in this configuration. And now I will pass the floor to Nicolas Vedren, who will share with us the future outlook in pharmacology and transplantation research. And yeah, we are very looking forward to hear your insights.
So good afternoon, everyone. I'm very pleased to be with you today and to share our current work and our vision for the future of pharmacology and transplantation research. So in this presentation, I am going to give an overview of how the field is evolving and how emerging technology, including, of course, organ-on-chip and also digital approaches, are reshaping our ability to predict patient outcome and personalize treatment. So for those of you who may not be familiar with Limoges, so we are located right in the center of the France. We say close to both the mountain and the ocean, at least if you have a car. But Limoges, it's a very pleasant city to live in, known for its iconic limousine core, but also for its beautiful train station, and of course, its world famous porcelain. But beyond that, Limoges is also home to excellent research laboratory, including, of course, our unit, and where we develop a fully translational research pipeline from computer modeling to bench and all the way to the patient bedside. So our goal is to advance translational science and contribute to the development of personalized medicine. So to give you a flavor of our team research, I'm gonna to tell you our story. So our unit was founded in 1995 with a strong focus on pharmacokinetic study in transplantation patients. So pharmacokinetic or PK is essential for the understanding the concentration of immunosuppressive drugs in each target organ and for optimize therapeutic exposure. So over time, the team developed innovative PK models that enable accurate AUC estimation and individuation assessment of drug behavior in transplant recipients. In the early 2000s, a large clinical and pharmacological database was created that is still widely used by clinicians and researchers. And this resource has become a cornerstone for transplantation follow-up, outcome prediction and disease modeling, and contributing to the improvement of graft management. Then, between 2005 and 2010, our research incorporated in vitro approaches, including metabolic studies on human liver microsome and drug transporter study using a human cell-based system. And as you can imagine, it was the old-fashioned before now the use of organ-on-chip. But this model allows us to characterize hepatic metabolism and investigate transporter-mediated process. From 2010 onwards, pharmacogenetics became an essential component on our works. By integrating the genomic information into PK, we were able to explain some inter-individual differences in drug and toxicity. Then in 2015, with the biomargin project, we began to investigate biomarker and kidney injury using proteomic analysis performed by high-resolution mass spectrometry. In parallel, in silico pharmacology became an important part of our research strategy. Computational model allow us to explore how drugs and metabolites cross biological membrane, interact with transporter, and distribute across tissue. So by combining molecular simulation and physico-chemical modeling and PK framework, we gain in a deeper mechanistic understanding of transmembrane passage and drug accumulation, for example. We further developed albumin focus analysis, which provided additional insight into early molecular changes associated with the liver function and helped refine the biomarkers identified always through HRMS. Now, in these recent years, we have also artificial intelligence has become a central pillar of our research. So machine learning model will help us to predict the appropriate doses and adjust the drug regimen according to each patient and individual profile. So more recently, we have explored also pharmacomicrobionic to understand how the gut microbiota influences drug metabolism, exposure, and also toxicity. It's important to know that microbial enzyme can activate or transform immunosuppressive compound, and microbial metabolite modulate host pathway relevant to graft health. So that is very important. And to finish now, we have advanced naturally converged toward our most recent focus, organ-on-chip and multi-organ-on-chip microphysiological system as described previously by Izzy. So today I would like to stop on a very important project which has taken a new dimension with AI and granted Major France 2023 award, the DIGFAT project led by Jean-Baptiste Waller. This funding allows us to develop a clinical and pharmacological digital twins capable of predicting individual treatment response in transplantation and beyond. The idea here is to combine clinical data, experimental readouts, synthetic data sets, and advanced PK/PD modeling into an unified digital framework. So these models are trained on anonymized patient data experimental pharmacology, and mechanistic simulation, and are then refined using artificial intelligence. So the goal is to recreate a virtual representation of each patient, a digital twin that can help clinicians anticipate drug exposure also and other therapeutic outcome. So that's true now that the field of pharmacology and drug development is going through a major change. So we have traditional approaches which mainly depend on animal models and static in vitro tests are now being replaced by dynamic human reference systems that combine for the in vitro part, for example, new stem cell model, but also organoids, and now we have the emergence of assembloid, and of course the multi-organ-on-chip. A key shift in the use of this technology, which mimic human physiology disease more accurately than ever before. So these advances reduce the need for animal testing and improve translational to the clinic. At the same time, machine learning method will help researchers map some adverse outcome pathway across many levels, from molecules and cells to organ and whole individual. So this multi-scale modeling approach supports the development of a virtual patient or digital twin that can simulate how compounds behave in different disease states, organ function, or combination, or different treatment. So the new vision of pharmacological is one where experiment data and model comes together to obtain a better, for example, personalized medicine, but also drug development, disease modeling, etc. So this is what we aim to develop within our unit in the coming years. By combining the expertise present in our laboratory, we plan to build a full pipeline of new approach methodology. This pipeline will include, of course, pharmacomicrobiomic, but also in silico analysis with, in the future, the ability to recreate virtual PK relevant cells. We also continue developing also organ-on-chip model, especially by extended towards some other applications than transplantation. Here it's a placenta on chip to study polluant. And we will mainly also maintain our focus on biomarker discovery with the support, of course, of artificial intelligence. We will be able to identify some very important key like the molecular initiating event. So our goal is therefore to develop a virtual twin NAM pipeline capable of predicting human effect by training on abandoned drugs or academic candidates and environmental pollutants. Ultimately, this NAMS-based virtual twin approach offers a transformative way to advance non-clinical pharmacology and toxicology. So it supports mechanistic discovery, improve human relevance, accelerate candidate evaluation, and expand our ability to assess novel also pollutant and complex exposure long before they reach the clinic. Sorry. So thank you for your attention, and I would like to thank, of course, all my team, and a special thanks like Izzy to Hanel and Antoine, who came to Limoges to help us with the OMI, so that was very great. And I want to add a special thank to Joseph and Jad, whom we met at the UOX conference in Milan in 2024. And our discussion, they are really what led to our presence here today. So thank you for the collaboration.
Thank you. Thank you very much, Nicolas, for the presentation and for the outlook on the pharmacology and transplantation and the research that is done at Limoges. Yes, we visited Limoges. It was very fun. It was great to see the lab. It's it's a very big lab with a lot of research and innovation going on. Thank you for welcoming us there as well. So let me continue on my screen here. So let's go on. I'll easy covered a little bit how Omi functions, but I can go into slight more detail to present Omi to the participants who do not know anything about it, about Omi system and how it functions. So here you can see the Omi portable system that can be controlled with a tablet, and it can maintain the cell culture inside the incubator for several weeks, and it allows the monitoring of the cell culture live, like this, like you can see in the picture. What is Omi? It's organ-on-chip platform that can control microphysiological fluidic environment inside the microfluidic chip. It performs long-term cell culture and the flow to generate ideal shear stress conditions. It's compatible with virtually any chip. So as you saw, the Easy used it with Beyond Chip, but also with the BD chip slides. You can control it with application on a tablet through Bluetooth or Wi-Fi, and it automates not only recirculation, but perfusion, injection, and sampling. The Omi itself consists of a touch screen that can be controlled manually in a remote mode. It has 4 reservoirs with liquid level sensors that allow to run the 4 distinct flow configurations. And here is the chip holder that can be used to place the glass slide sized chip. It allows the perfusion, so you can see the reservoir on top, the RI and RO represented in the schematics here. So the perfusion is the perfusion from one reservoir to another. After a small volume perfusion, you can collect the media for conditioning or for assessment. There is also the unidirectional recirculation that allows to recirculate the medium from reservoir RI to RO. And this can be done for several days or weeks, depending on the experimental setup. Then there is an injection reservoir I that can inject, you can inject the drug of interest in a controlled way inside your fluidic circuit. And there is a sampling reservoir. It is only one reservoir, so it can accommodate only one sampling if necessary during night. I'll present an example of vessel-on-chip validation that we used internally at Fluigent over the 5 days of cell culture of HepaRG to mimic the blood vessel-on-chip with Bflow microfluidic standard chip. We daily monitored it under microscopy for cell behavior, morphology, and growth division. It is a qualitative assessment of the cell viability. And it will allow me to introduce the classical workflow you can do with Omi system. So here we have the first day we seed the cell with a fibronectin coated surface with HepaRG with a given number of cells. And on the day 2, after leaving the culture overnight, we set up and calibrate the Omi device. As Easy mentioned, Omi takes some time to calibrate and set up. So it could be 1.5 to 2 hours, obviously not including all the chip preparation that is done in advance. Then the device is connected for media recirculation. In our case, after 4 hours at 50 microliters per minute for cells to acclimate, we change the medium to remove the non-adherent cells from the recirculation and continue the recirculation for 5 days, gradually increasing the flow rate from 100 to 200 to 300 to 400 to 500 microliters per minute. The recirculation for 5 days allowed us to have the small amount of medium used in the our protocol, and to monitor the cells on the chip daily. The interesting thing that we observed is that there is a top layer colonization of endothelial cells under flow conditions. So this type of behavior is not happening in the static environment and is observed after 3 days when we increase our flow rate to 300 microliters per minute, which is equivalent to 1.37 dyne. And it was observed first on the day 3, so of cell culture, and by day 7 of recirculation, it reaches the confluency. Another interesting thing that we found is that at day 1, our chip looks like something like this, with quite a lot of dead cell and debris precipitation, even after 24 hours. And then after 3 days, you already can see that endothelial carpet is quite clean, with cells starting to elongate. Under the seventh day of the flow, with a high flow condition, cells align to the direction of the flow. They migrate within the chip to the top channel. So the colonization and vessel-like structure formation happens. It exhibits increasing spacing in between the cells after a higher shear stress condition. It also could be due to the detachment of the cells under the high stress. And it reaches confluency both on the top and the bottom levels. So we illustrate this with the HepaRG cells because they're most sensitive to flow, and it allows us to investigate the flow control of Omi on the microfluidic chip. So to conclude, Omi is a versatile platform that allows the 4 distinct functions. It's compatible with any chip. It reduces contamination by using the disposable cartridges, but also with including the sterilization protocol, allowing an extra disinfection step in your workflow. It's compact and autonomous. It works on the battery for 3 to 4 hours. It allows a remote control via tablet, and it's compatible with existing biological infrastructure such as incubator and the microscopes. And with multiple Omis, you can also gain in productivity. So having 3 Omi for 3 experimental conditions, or as I showed, having it in the dual configuration. To end this webinar, I want to shortly introduce Fluigent, our company. At Fluigent, we help researchers and industries to be successful in their application of fluid control at a micro scale, where it is critical. We provide thousands of flow controllers worldwide with a subsidiary presence in US, but also over 10 distributors around the world. Our products have been widely used in top research laboratories, and we are also quite present in France. And for more than 10 years, we're featured since 2009. It's already 15 years. We have more than 3,000 publications with Omi and with Fluigent instruments, which here you can see is the Flow-Easy. And Fluigent pressure controllers are cited in these papers. So with that, I would like to thank our guests for joining. Thank you, Nicolas and Izzy, for presenting as well. Thank you, the participants, for your attention. I hope you enjoyed the webinar. And here is the end of the webinar. However, we have a Q&A session afterwards that is not going to be present in the replay. And finally, I have to add 2 points. You can contact, if you have any questions, contact@fluigent.com, but you also can have here my professional email to reach out if you have any questions about Omi or your organ-on-chip application. And you can also, from me, request an Omi demo.
Thank you very much.
Yeah, have a good afternoon. Goodbye.
See you.
Bye. See you.